Thank you very much. I apologize that I can't be there in person, but life has been complicated lately. I'm very happy to talk and introduce this seminar series. I think what's critical to understand is that Katie and I started this work 27 years ago. And from the beginning, my goal has always been to include the entire world, to allow equity across the world, to give access to science, to mRNA, to vaccines, to therapeutics. And with Afrigens and the WHO and many other groups' help, we've been able to accomplish this. So the mRNA revolution, which actually started quite a years before the COVID-19 vaccines came out, has mostly centered on vaccines. And the wild success of the COVID-19 vaccines, about 95% efficacy in phase 3 trial, show the power of this vaccine platform. It's currently being used for many different diseases, many different infectious diseases, from viruses like HIV, hepatitis C, norovirus, Zika, other flaviviruses, many different viruses. But there's also applications in cancer. We've seen a couple of results from clinical trials showing success in melanoma and pancreatic cancer. And new developments involve making vaccines for autoimmune diseases and food and environmental allergies. So the wide-ranging applications of the RNA technology are really enormous. And this is something that the whole world needs to do. My thinking of this is that regions have diseases that are specific for their areas. In order to allow local regions to develop therapeutics they need, they have to have access to the RNA scientific technology and to the ability to produce GMP vaccines, which is what's been accomplished through this WHO program. The future of RNA is also quite enormous. It's currently being used as a therapeutic protein delivery system. So we and others have been delivering RNA to the brain, to the heart, lungs, T cells, bone marrow stem cells, and many others. And the big difference here is that current gene therapy approaches involve taking cells out of a person, putting them in culture, infecting them with a virus that encodes gene editing technology that fixes the gene defect, and then putting those cells back in. Such a therapy is about to be approved by the FDA in the United States for sickle cell. The problem is that this requires extensive clinical and research facilities that are not available across the world, and it's likely going to cost in the region of $1 to $4 million a person. When you've got 300,000 people a year being born with sickle cell anemia, the numbers don't add up. So the approach that we've been taking is in vivo gene therapy. And what that means is that we use RNA LNPs and we target the LNPs to the cells of interest. We've been able to deliver these LNPs to bone marrow stem cells and deliver gene editing technology. So we can fix the defect in sickle cell anemia in the body with a simple IV injection of off-the-shelf RNA LNPs. That can be easily given across the world by a simple intravenous injection. This has wide-ranging applications to thousands of other genetic diseases of the bone marrow, as well as diseases of the liver, lung, brain, heart, and many other organs. Therapeutic protein delivery was actually one of the earliest developed technologies. Currently, there are phase 2 going into phase 3 trial using RNA to treat heart attacks and heart disease. We see this as a rapidly expanding area of research. Heart disease is the #1 killer in the world, so it's incredibly important for the entire world. By having GMP sites across the world that can produce these therapeutics and develop their own therapeutics, it opens up the possibilities for new RNA therapies. So to me, this is an incredibly exciting moment in the field of RNA research. And the development through WHO, who funded and helped Afrigen build their GMP site, I think is going to be critical to allowing wide-scale RNA use in the future. Thank you very much.